Hello, everyone. Good morning. Hope you are all doing well. Welcome to the last day of our International Symposium on Biological Sciences. So, to start the section today, we have Professor Tiago Januário da Costa. He is a biologist, master and PhD in biological science at the focus in pharmacology at the Institute of Biomedical Science of the University of São Paulo in Brazil. The main projects focus in the functional and molecular study of the mechanism of action of sex hormones, especially estrogen and testosterone, on the cardiovascular system in models of postmenopause, aging, and arterial hypertension. He's a visiting researcher at the Department of Pharmacology, Therapeutics, and Toxicology of the Universidade Autônoma de Barcelona, and at the Instituto de Investigações Biomédicas Auguste P. Sunier at the Universidade de Barcelona, participating in basic and clinical science projects aimed at seeking the effect of DNA methylation, alternative splicing of estrogen receptors, and microRNA in the development of cardiovascular disease in postmenopausal women. So, Tiago, thank you for accepting being here with us today, and welcome. Please, you can start your presentation. Thank you.
Thank you. Good morning, everyone. I start my presentation. So, thank you so much for invitation to present my work and that all collaborators. It's a big pleasure to be here and to talk about SARS-CoV-2 and cardiovascular system. I don't have conflict of interest in this presentation. So, the endothelial cells is a monolayer cells present in the lumen of all vessels and promote a crucial interface between blood and different tissues. Under physiological condition, endothelial cells promote vasodilator or vasoconstriction, promote antioxidant, increase the antioxidant system, anti-inflammatory response, and control the vascular permeability. In the physiological injuries, for example, high blood pressure or atherosclerosis, the function of endothelial cells change and induce the cardiovascular disease. It's very interesting that the cardiovascular disease is a common comorbidity present in COVID-19 patients. In this study, suggests that 15% of the patients with COVID-19 present hypertension and 13 present coronary artery disease. In addition, during the hospital stay, 32% of the COVID-19 patients need to vasopressor support to control the blood pressure. In addition, this study suggests that 4 important biomarkers of cardiac injury was associated a death in COVID-19 patients. So, these results suggest that an important correlation with cardiovascular system and SARS-CoV-2 infection. Several studies suggest that the cardiovascular system, specifically the endothelial cells, changes after the SARS-CoV-2 infection. The first study showed the blood flow decrease in COVID-19 patients, and this increased the risk of thrombosis. In next talk, my colleague Simone will talk about how the plasma from COVID-19 patients induced the degradation of important proteoglycans present in the endothelial cells. So, the SARS-CoV-2, the coronavirus, the spike protein present in coronavirus is responsible to attachment in the ACE2 present in the host cells. After this bind, a specific protease, TIMP2, cleave the spike protein and contribute the fusion of the virus in the host cells. After this, the virus enter in the cytoplasm and uncontrolled SARS-CoV-2 infection initiated and induced cytokine storm, whereby pro-inflammatory response over produced by immune system. This phenomenon induced end-organ disease, including the cardiovascular disease. It's very important to mention that this cytokine storm is mediated by toll-like receptors. The humans present about 10 toll-like receptors, and in 2019, Ashen and co-authors suggest that the mitochondrial DNA induced toll-like receptor activation and cardiovascular dysfunction. Their data showed the spontaneous hypertension increased the mitochondrial DNA compared to normal intensive rats, and in turn, under adrenergic stimulus, mitochondrial DNA increased interleucin 6 and TNF-alpha, and this increase induced vasoconstriction. It's very importantly that the OGN2088, a toll-like receptor antagonist, decrease the vasoconstriction induced by mitochondrial DNA. So, this result suggests that the mitochondrial DNA is important agonist to toll-like receptor 9, and this receptor induces the vascular dysfunction. To explore this idea about SARS-CoV-2 and cardiovascular disease mediated by toll-like receptor, the hypothesis of my work is the SARS-CoV-2 infect endothelial cells, promote the mitochondrial dysfunction, and increase mitochondrial DNA release. In turn, mitochondrial DNA induce toll-like receptor signaling and an important downstream to induce the inflammation. To explore this hypothesis, we used UVAC cells infected by SARS-CoV-2 for 48 hours, plasma from COVID-19 patients and respective controls, and for functional studies, we used mice. So, my first question is, does SARS-CoV-2 infect endothelial cells? For this, we evaluated the protein levels to ACE2 and TIMP2, two important key to infect the cells, and we observed that both ACE2 and TIMP2 are robustly expressed in endothelial cells. In fact, using the specific antibody to detect SARS-CoV-2, we observed that the presence of the virus in endothelial cells. However, this infection is lower compared to positive control, the viral E6 cells. It's very interesting, it's very important to talk about the SARS-CoV-2 infect endothelial cell, but does not replication in endothelial cells. So, the next question is associated to SARS-CoV-2 and the mitochondrial function. Using mitosox-red, a specific superoxide indicator in mitochondria, we evaluated the regeneration in mitochondria. In this channel, we observed the representative image, and the graphic, we observed that the inactivity and SARS-CoV-2 infection increased the fluorescence to mitosox compared to control mock. We observed in the same in the representative images. In this experimental protocol, we used rotenona as a positive control because the rotenona is a strong inhibitor of complex 1 of the mitochondria, and the inhibitor of complex 1 increase the Rho generation. It's very interesting that SARS-CoV-2 decrease the protein levels of complex 1 compared to control mock, suggesting that it is a possible mechanism that increases the Rho generation in SARS-CoV-2 infection. In next step, we evaluated another mark for mitochondrial dysfunction, is a membrane potential by mitotract red probe, and we observed that the SARS-CoV-2 infection increased the fluorescence by mitotract compared to inactivity virus in mock control. The same was observed in the representative images. In the mitochondria, the VDAC, or voltage-dependent ion channel, is an important channel to control the membrane potential in the mitochondria because this channel regulates a flux of different ions, including the calcium. And it's very interesting that SARS-CoV-2 decreased the protein levels of VDAC compared to MOC, suggesting that this increase of the potential of membrane was associated to low levels of the VDAC channel. In fact, we tested the role of the calcium in endothelial cells and with a specific probe to detect the cytosolic free calcium, and we observed that in UVEX infected by SARS-CoV-2, the response of calcium is low compared to inactivity virus and control mock, suggesting that the changes in VDAC channel contribute to reduce the response to calcium in endothelial cells. In addition, we evaluated the ATP induced calcium in endothelial cells. However, this phenomenon was abolished in the cells infected SARS-CoV-2. So, these results suggest that all mechanisms to control the calcium in endothelial cells was abolished in the presence of the SARS-CoV-2. The next step, we evaluated if that the mitochondrial dysfunction contribute the mitochondrial DNA release. First, we evaluated the integrity of the membrane plasmatic, and we observed that the SARS-CoV-2 infection increased the degradation of membrane compared to respective control, and the toll-like receptor antagonist, the OGN2088, decreased the membrane plasmatic degradation. It's very interesting because this in the UVEC cell infected by SARS-CoV-2 increased both marks of the DNA mitochondria compared to respective control. In addition, we observed the same phenomenon in the patients infected by SARS-CoV-2, patients with COVID-19, compared to comorbid patients and compared to health patients. It's very interesting result because it suggests that the SARS-CoV-2 induced mitochondrial DNA release in vitro studies and the in vivo studies. So, the first part of this study demonstrated that the SARS-CoV-2 infect endothelial cells and promote the mitochondrial dysfunction because increased Rho generation and membrane potential. This phenomenon increased mitochondrial DNA release. And my next step, my next question, I evaluated if mitochondrial DNA induced toll-like receptor 9 activation. First, we evaluated the protein levels of toll-like receptor 9, and we observed that the SARS-CoV-2 infection increased the protein levels of toll-like receptor compared to respective control, and in addition, we observed that the inflammatory response mediated by NF-kappa B was higher in SARS-CoV-2 compared to respective control. We evaluate the total and the phosphorylate NF-kappa B. It's very interesting that the antagonism of toll-like receptor 9 decrease the protein levels if total and phosphorylate NF-kappa B, suggesting that the toll-like receptor 9 mediated the inflammation. In fact, we evaluated the levels of EL6, and we observed that the SARS-CoV-2 increased the levels of this interleucin compared to respective control, and toll-like receptor 9 antagonism decreased the production of IL-6. We evaluated too the production of thromboxane and endothelin because there's a two important vasoconstriction, and the result is very interesting because the SARS-CoV-2 infection increased the thromboxane production and decreased endothelin production. However, both changes is not dependent of the toll-like receptor agonist receptor. Last, for evaluated the role of mitochondrial DNA in vascular dysfunction mediated by toll-like receptor 9, we evaluated by vascular reactivity the aorta function of the mice, and we observed that the incubation of mitochondrial DNA induced vasoconstriction compared to respective control and positive control with a genomic DNA. It's very interesting that this vasoconstriction was abolished in the toll-like receptor 9 knockout mice. So this result suggests that the release of the mitochondrial DNA induced vascular dysfunction mediated by toll-like receptor 9. So, in conclusion, these results suggest that the SARS-CoV-2 infects endothelial cell but does not replicate it, induced mitochondrial dysfunction, and mitochondrial DNA release. In turn, mitochondrial DNA release induced toll-like receptor activation, increased the production of IL-6, and induced endothelial cell dysfunction. I would like to thank all group involved in this project, especially the postdoctoral fellow in Ribeirão Preto Medical School. I would like to thank Professor Rita Tosse in Ribeirão Preto Medical School and her group. I would like to thank Professor Natasha Zahara in Johns Hopkins School of Medicine and your group. And thanks so much for FAPESP, CAPS, and CNPq for financial support. Thank you for your attention, and open the questions in Portuguese or English, no problem.
Okay, Tiago, thank you for pleasing us with such a great results. And we know that talking is faster than typing. So while people take a little while to type the questions, I would like to make one. Oh, sorry, we have two questions here already. So Professor Diego Colunati is asking why you try to investigate different variants of SARS-CoV-2. And Dr. Carlos Castro is asking, he's congratulating you on a very nice presentation and asking, is there some explanation for why SARS-CoV-2 does not replicate in endothelial cells?
So the first question, I don't investigate another variant because I started this project in the first variant, but it's a very good point that the second variant induced the same dysfunction, but I don't evaluate. This is the first coronavirus, the first sequence of coronavirus. The second question, I don't see the second question. Explanation is a very good question about the replication endothelial cell. In fact, I don't know. I have a different hypothesis. There are a preprint about the SARS-CoV-2 needs the glutamine to replication, and in UVEC cells, the glutamine concentration is low. It's a possible to do replication in UVEC cells, but I need to more investigation to understand the physiological condition of endothelial cells that contribute to stop the replication of the virus because it's a specific endothelial cells. So my first hypothesis is associated to a glutamine, but I need more investigation about this.
Okay, Tiago, I have a question.
Yeah.
I've seen a lot of hypotheses on the role of renin-angiotensin system in the process, the pathophysiology of SARS-CoV-2, and starting with the ACE2, the connection of the ACE2 with the spike protein and all this stuff. So do you know whether already there is a bunch of data showing that patients taking drugs interfering with the renin-angiotensin system like losartan or captopril, if this interfered with the pathophysiology of the disease?
It's a very good question. In most cases, the use of antihypertensive treatment inhibit the ACE1 enzyme, no ACE2. I think that is an important correlation. Is the virus enter in the cells by ACE2 and developed independent of pharmacological treatment. In most cases, for example, in studies, we showed all patients used antihypertensive treatment and the progress the disease is the same. So I think the pharmacological treatment is important to control the blood pressure, but don't inhibit the virus enter in endothelial cells and contribute the dysfunction.
So it looks like ACE2 is the single point of the system which is involved.
Yes,
okay.
Yes, the single point.
Okay. We have more questions? We have time for more questions?
Wow, yeah, I have many time.
So I have another question, Tiago, about the involvement of the nitric oxide and reactive oxygen species. So you showed a lot of endothelial data, and what would be the involvement of these two molecules in the pathophysiology of the disease in endothelium?
Yes, we will evaluate the protein levels of enos, and the enos decrease and consequently decrease nitric oxide production. I think this is another mechanism to increase vasoconstriction. I think that in next talk, Simone talk about the nitric oxide production in the endothelial cells treated with plasma from COVID-19 patients, but I think that the nitric oxide is involved in the vascular dysfunction too.
So do you have access to any data showing that the antioxidant therapy changed the course of the disease?
It's a very good point. I don't read about, but it's a very complicated because I study aging, and all the studies, the antioxidant treatment doesn't inhibit aging because they are a physiological increase the loss important to control the vascular tonus for the aging process. I don't know. I think that is antioxidants is a not good treatment for impact the virus infection.
So it's not going to change the surveillance or something.
Yeah.
Okay. We have another question from Dr. Fernanda Giacchini. Tiago, how was the experience of working with COVID-19 during the pandemic?
Well, very complicated because I stopped my postdoc project and I will start work with COVID-19. I don't understand about virus. I understand about this cardiovascular disease. So I start the new topics in my career I changed the country. I started the postdoctoral in United States. In the pandemic is very complicated because all universities closed. So it's a very complicated, but I need to change my focus in this moment to respond this global problem, but it's a very complicated.
Sure. Looks like Dr. Fernanda has another question. African Americans were much more committed by COVID-19 in USA. Anything related with variations of endothelial cells?
Oh, Fernanda, is a very good question. In this moment, in United States, talk about all the time with the cardiovascular disease in America, in African America, and white people. Aqui, in United States, talk white people and black people. It's very interesting because the black people present a high blood pressure compared to white people, and this is important to increase the risk of the coronavirus infection or developed consequences after the SARS-CoV-2 infection. I don't know about the ACE2 variant or virus variant infection difference black people and white people, but the cardiovascular system is very different, and because of this, I think the development of disease is very different. But it's a very good point. It's a very good point.
Tiago, I got a last question. It's a technical question. What is the risk of your infection dealing with the samples in the bench?
No, I worked in a specific laboratory, safe number 3, with a different mask, different clothes. Is a specific. I developed all experiment in this laboratory.
Okay, hope you stay healthy.
Yeah. Yes.
So, guys, we have more questions. Okay. So, Tiago, once again, thank you for being here with us today, and very nice work. And it's very relevant for our future.
Thank you.
Thank you for this as well.
Thank you.
Once again, thank you. We can proceed with the next presentation.
Okay. First of all, I would like to congratulate the organizing committee for the excellent choice of the speakers. It's a pleasure to participate together with the Professor Carlos Xavier as coordinator of the session. The next lecture will be given by the Professor Simone Regina Potje, who is a biologist and earned both master's and PhD degree in physiological science at the State University, at São Paulo State University, Unesp, Júlio de Mesquita Filho, by the Multicentric Graduate Program, Programa Multicêntrico de Ciências Fisiológicas da SBFIS. She spent a period at the University of Illinois during her PhD. Later, she was a postdoctoral fellow at the University of São Paulo, with periods of visiting scholar at the University of Illinois and at the University of Arizona, at Tucson. She had also a researcher position at the Indiana University, and currently she's a professor at the State University of Minas Gerais, Universidade Estadual de Minas Gerais. Professor Simone works in the area of cardiovascular physiology and pharmacology. We focus on how physiological and pathological situations alter vascular cells. The title of her lecture today is Glycocalyx Degradation is Induced by COVID-19. So, Professor Simone, thank you very much for your availability to participate in this event. So, I will give you the floor.
Good morning to everyone. Professor Aline, can you hear me?
Yes, I can.
Yes? Okay. I will share my screen. Can you see my screen? Everyone can see?
Yes.
Yes.
Perfectly.
So, good morning again. As Professor Aline told, my name is Simone, and currently I'm professor at Minas Gerais State University. And the title of my work today is Glycocalyx degradation is induced by COVID-19. So this research was performed in Ribeirão Preto Medical School in the last year under supervision of Professor Rita Tostes, and I was doing my postdoc in University of São Paulo. And these are the co-authors of this work, and Tiago Costa, the first speaker, worked in this research together with me. So here, let's put the pointer here. Yes. So here we can see a blood vessel, and we can see vascular smooth muscle cells. Above vascular smooth muscle cells is a layer of endothelial cells, and also above endothelial cells we have the glycocalyx. So in this image we can see better the glycocalyx. Here is endothelial cells and the cavalvular structure, and we can see the glycocalyx. So the glycocalyx is glycoproteins that stay attached in endothelial cells, and is composed mainly by heparan sulfate chains. In the red color, we can see the heparan sulfate chains. And the protein is attached to heparan sulfate chains and is inserted in endothelial cells. So we can see the glypican 1, syndecan 1, syndecan 2, 3, and 4 compose the glycocalyx layer. Also, the glycocalyx is composed by hyaluronic acid in the green color, as we can see in this image. And the glycocalyx has a lot of functions in the vascular field. Glycocalyx can act as a barrier and selective filter. The glycocalyx work as a protector of vascular cells. Also, glycocalyx prevents leukocyte adhesion, avoid the antithrombotic events. Also, the glycocalyx is pro-angiogenic, and the main function that I have been working with the glycocalyx is about the mechanotransduction. So what is the mechanotransduction? The mechanotransduction is when the glycocalyx work as a sensor of the shear stress. So when the cells of the blood, or when the diameter of the vessel decreases, increasing the blood pressure values, the shear stress increases, and the glycocalyx can sense this, can work as a sensor, and the glycocalyx activate nitric oxide production by activation of endothelial nitric oxide synthesis. So the glycocalyx mediates the mechanotransduction. Since the glycocalyx has a lot of important functions in the vascular field, when the glycocalyx is not present, is degraded, we have a lot of problems in the cardiovascular vessels, the vascular vessels. So when we have the disruption of the glycocalyx, because its degradation, we have a loss of coagulation control and activation of thrombotic events. We have a loss of antioxidant defense, since the superoxide dismutase stay attached to the glycocalyx, to the glycoproteins in endothelial cells. Also, a degradation of the glycocalyx compromise the leukocyte and platelet adhesion. Also, the disruption of the glycocalyx promotes edema formation, and also we have an impairment of the mechanotransduction. Then the vasorelaxation is impaired. But what promotes the degradation of the glycocalyx? So when we have increased levels of cytokines, and also when we have an increased level of reactive oxygen species, both cytokines and reactive oxygen species can activate heparinase. So heparinase is an enzyme that cleaves heparan sulfate chains, and as I told you in the beginning, heparan sulfate chains is the main protein present in the glycocalyx. So when heparinase is activated, cleave this heparan sulfate chains and promote the glycocalyx degradation. So the hypothesis of our work is that plasma of patients with COVID-19 promotes glycocalyx degradation. In the first talk of Tiago, he showed his results and he used the virus, the SARS-CoV virus in his experiments. But in my work, we did not use any virus. We used just plasma from COVID-19 patients, and also we used the endothelial cells, HUVEC, and we treated the cells with plasma from COVID-19 patients. So here is my hypothesis. And what is the ends of my work? So the ends of this study was to evaluate if COVID-19 promoted glycocalyx shedding, to determine whether plasma from COVID-19 patients induce glycocalyx degradation, and also to investigate if heparin avoids the glycocalyx disruption. Why I use the heparin? Heparin is an anticoagulant, and also heparin inhibits the heparinase activity, and heparinase cleavate the heparan sulfate chains. Then we used in this work plasma from COVID-19 patients and health subjects. This is the ethics committee protocol number. And to check if the health subjects did not have COVID-19 disease, we made the test of the IgG and IgM antibodies in these patients. And as we can see in this image, all 7 healthy subjects were negative to IgG and IgM antibodies. And to patients with COVID-19, we did the PCR to check, to confirm the SARS-CoV infection. Also, we did the detection of the specific antibodies IgG and IgMs that were positive to all patients with COVID-19. And also in this study, we used health subjects, men and women, and we used men and women, similar numbers of men and women with COVID-19 disease were used in this study. And in our analysis, we split the COVID-19 group in mild to severe and a critical disease of COVID-19 patients. And how we split this group? Because we checked the rate of oxygen that is an indicator of lung injury, and also we checked the dime levels that indicate the gravity of this disease. Then we performed ELISA assay. Also, we performed Western blot assay. We used the fluorescence probes to check the nitric oxide levels and hydrogen peroxide levels. And we did the immunofluorescence assay to confirm our results. So in the first result, I used just plasma from COVID-19 patients and from health subjects. Here, I don't have endothelial cells yet, just plasma from the patients. So we checked in the plasma, we checked the levels of interleukin 6, interleukin 1 beta, and we checked the TNF alpha. And as we can see, plasma from COVID-19 patients was increased the interleukin 6 levels compared to health subjects, and also the interleukin 1 beta was increased in plasma from COVID-19 patients compared to health subjects. When we split this group and between mild to severe and the critical, we did not observe any difference between levels of IL-6 and IL-1 beta. And when we checked the TNF alpha levels, we did not observe any difference between COVID-19 patients and health subjects. Also, when we split this group between medium to severe and the critical, we did not observe any difference. And when we checked the stress oxidative through T-bars assay, we observed that T-bars level, MDA levels, were increased in plasma from COVID-19 patients compared to health subjects. Also, we did not observe a difference between this group when we split in mild to severe and the critical. So these first results demonstrate that plasma from COVID-19 patients is altered. So exhibit increased cytokine and oxidative stress compared to health subjects. So the plasma are already changed, the plasma from COVID-19 patients. So in our second step, we also used the just plasma from this health subject and COVID-19 patients, and we checked the human heparinase activity. Remember that heparinase is the enzyme that degrades, cleavates the heparan sulfate chains that is the main component of the glycocalyx. So when we checked the activity of this heparinase enzyme, we can see that the activity was increased in plasma from COVID-19 patients compared to health subjects. But when we split this group between mild to severe and critical, we did not observe any difference. Also, in the plasma from these patients, we analyzed the heparan sulfate proteoglycans, and we can observe that heparan sulfate proteoglycans is increased in plasma from COVID-19 patients compared to health subjects. And we did not observe any difference between mild to severe and the critical patients. So since heparan sulfate proteoglycan is increased and also heparinase activity is increased, these both results indicate that the glycocalyx of COVID-19 patients is being degraded, is being shedding. And also we analyzed the HRL1 levels, that is another component of the glycocalyx. As we can see, also the HRL1 levels were increased in plasma from COVID-19 patients compared to health subjects. But when we split this group between mild to severe and the critical, we did not observe any difference. So plasma from COVID-19 patients exhibited an increased glycocalyx components compared to health subjects, indicating the glycocalyx shedding. So in the next step, we plated the health endothelial cells, human umbilical vein endothelial cells. We plated it in 96 wells, and we treated the cells with plasma from COVID-19 patients and plasma from health subjects. So when we treated the HUVEC cells and we analyzed the hydrogen peroxide using the probe 82DCFA, and also when we analyzed the nitric oxide levels using the probe DAF2DA, we can observe that both hydrogen peroxide and nitric oxide levels were decreased in HUVECs treated with plasma from COVID-19 patients. So there is a difference. When we split the COVID-19 group, we did not observe any difference between mild to severe and the critical for hydrogen peroxide, but we observe increased levels of fluorescence probe DAF2DA to critical patients compared to mild to severe. Also, we checked the reactive oxygen species. So we use the probe D8E, that is non-specific, but we used it as a specific to anion, superoxide anion. And also we use the 7CBA, that is a specific probe to peroxynitrite. When we check the reactive oxygen species using this both probe, we did not observe between difference in HUVECs treated with plasma from health subjects and treated with endothelial cells treated with plasma from COVID-19 patients. As we can see, no difference. Also, when we split this group between mild, severe, and critical, we did not observe any difference. But since we did not observe any difference here in reactive oxygen species levels promoted by a plasma from COVID-19 patients, but we can observe difference here. So plasma from COVID-19 patients changed the vascular homeostasis of HUVEC since it changed the levels of hydrogen peroxide and nitric oxide levels. Then these results indicate that plasma from COVID-19 patients decreases nitric oxide and hydrogen peroxide in endothelial cells. And in the next step, since plasma from COVID-19 promoted this decreased fluorescence in this both probe, we want to check if our treatment with plasma from COVID-19 patients were impair the endothelial cells function. So we performed the treatment for lactate dehydrogenase, that is indicative of tissue damage. And when we treated the HUVEC with PBS was our control group, and when we treated the HUVEC with plasma from healthy subjects, and when we treated the HUVEC with plasma from COVID-19 patients, we did not observe any difference in LD8 levels. And here is our positive control. So the treatment with plasma from COVID-19 patients did not promote any tissue damage or any cell damage. And also, we use the supernatant of these cells, and we check the heparan sulfate levels. And as we can see, the plasma from COVID-19 patients increased the heparan sulfate levels in the supernatant of these cells compared to a treatment with plasma from healthy subjects and the treatment with PBS, our control group. So this demonstrated that the plasma were promote the glycocalyx shedding in HUVEC cells. So plasma from COVID-19 patients promote the glycocalyx shedding in healthy endothelial cells. To confirm this result, we performed the Western blot assay. So we plated HUVEC cells in 6 wells plate, and we treated the cells with plasma from COVID-19 patients and plasma from healthy subjects. And we performed the treatment for 30 minutes, 1 hour, and treatment for 12 hours, and treatment for 24 hours. And as we can see, we treated also HUVEC with a PBS that was our control group. And as we can see, when we treated the HUVEC with using plasma from COVID-19 patients, decreased the expression of Syndecan-1, of Glypican-1, and Syndecan-4 with 30 minutes of treatment, and also with 1 hour of treatment. So the plasma from COVID-19 patients was effective to promote the glycocalyx shedding, since decreased the expression of these specific components of the glycocalyx. Also, the treatment for 12 hours and 24 hours were more expressive, as we can see, decreased the expression of Syndecan-1, Glypican-1, and Syndecan-4 compared to treatment of HUVEC with plasma from healthy subjects and our control group, the PBS. Also, we can see here that the plasma from COVID-19 decreased the expression of these specific components of the glycocalyx. So these results demonstrate that the plasma from COVID-19 patients decreased expression of specific glycocalyx components. And in the next step, since heparinase was increased in the plasma from COVID-19 patients, and heparinase is responsible to cleave the heparan sulfate chains, we want to check if heparin could avoid the glycocalyx shedding, the glycocalyx degradation. Then we used the HUVEX, and we treated the HUVEX with plasma from health subjects, and we treated the HUVEX with heparin, low molecular weight heparin, and we treated the HUVEX with plasma from COVID-19 patients, and also we treated the HUVEX cells with plasma from COVID-19 patients in presence of heparin, with low molecular weight heparin. And we analyzed the same proteins that we analyzed here. So as we can see, syndecan-1, glypican-1, and syndecan-4. And only the treatment with plasma from COVID-19 patients decreased the expression of syndecan-1, glypican-1, and syndecan-4, as we can see in the graphics below. So here, decreased the expression of syndecan-1, increased the expression of glypican-1, and decreased the expression of syndecan-4. But heparin treatment was effective to avoid the glycocalyx shedding. So in presence of heparin, we did not observe a change in syndecan-1 levels, syndecan-1 expression, or glypican-1 expression, or syndecan-4 expression, as we can see here in this group. So these results demonstrate that the treatment was with heparin was effective and avoid the glycocalyx degradation in health HUVEC endothelial cells. So to confirm these results here, we did the immunofluorescence assay. And in this immunofluorescence assay, we used the glypican-1 antibody, because the glypican-1 is mainly expression in endothelial cells. And since we were using HUVEC endothelial cells, so we choose to perform immunofluorescence for glypican-1, just for glypican-1. And in the first line, we have HUVEC treated with PBS. In the second line, we have HUVEC treated with plasma from health subjects. And in the third line, we have HUVEC treated with plasma from COVID-19 patients. And in the last line, we have the HUVEC treated with plasma from COVID-19 patients in presence of heparin, of low molecular weight heparin. And as we can see, here is our the nucleus of the cells. Here is the mark for endothelial cells. Here is our target, the glypican-1. Here we merged the endothelial cells with the nucleus. And here we made a zoom of these specific areas, as we can see. And the treatment with plasma from COVID-19 patients decreased the fluorescence for glypican-1, as we can see in the image, and we can see in the graph here. And when we did the treatment with heparin and also plasma from COVID-19 patients, heparin was effective to avoid the decrease of fluorescence, as we can see. So heparin avoid the decrease of immunofluorescence for glypican-1. So these results confirmed the results that we demonstrated here, that the heparin is effective to avoid the glycocalyx shedding. Then, taken together, these results demonstrate that plasma from health subjects did not demonstrate, in the beginning of the experiment, did not demonstrate oxidative stress or interleukin levels in plasma from health subjects. So when we treated health endothelial cells with this plasma, endothelial cells presented a redox balance and a regular inflammatory cycle. Also, the glycocalyx remained intact, and the endothelium function was kept during the treatment, since we did not observe difference in nitric oxide levels or hydrogen peroxide levels in endothelial cells treated with plasma from healthy subjects. But when we treated the HUVEC cells with plasma from COVID-19 patients, we observed in the beginning of the experiments that the plasma from these patients presented oxidative stress, also presented the interleukin 6 and interleukin 1 beta increased the levels. And when we performed the treatment of healthy endothelial cells with this plasma, we observed a redox disbalance, since the plasma from COVID-19 patients decreased the nitric oxide levels and decreased the hydrogen peroxide levels. Also, we observed that the plasma from COVID-19 patients promote glycocalyx degradation, glycocalyx shedding, and this reduced the endothelial dysfunction. And when we used heparin, low molecular weight heparin, we observed that avoided the glycocalyx shedding and the glycocalyx degradation. So this is our conclusion. And I want to thank all co-authors that helped us to perform these experiments. I want to thank the lab toasts students, and I want to thank the organization of International Symposium on Biological Science to invite me to make the presentation today, especially Professor Manuel Biancardi that made the invitation to me. And I want to thank you the financial support, CAPES, CNPq, and FAPESP. And this is my contact if someone has questions after or want to make contact and some collaboration. So thank you for everyone.
Thank you very much, Professor Simone, for this enlightening lecture. Now we are open to questions. We have a question from Larissa Matuda Macedo. She asked, congrats, Simone and Tiago. Did your group study these mechanisms in diabetic patients with plasma from diabetic patients?
So thank you for the question, Larissa. We did not use the plasma from diabetic patients, but now I think in this moment in Tosta's lab, there are some students using plasma from obesity subjects and using plasma from diabetic patients, doing both experiments to check if there is some difference not in the glycocalyx, but in other mechanisms.
Mm-hmm. And we have another question from Professor Diego Basilico Lugnati. In the beginning, we have seen huge expectations about the convalescent plasma treatment for COVID-19. Based on your data, do you think that it could be an issue?
I don't know, because thank you for your question, Professor Diego. I read about the convalescent plasma, but there is a lot of studies in this moment in Brazil and in the world using convalescent plasma, but I don't know if it's a good idea. I think we need to study more, and I think the SARS-CoV study is in the beginning in the world. I think we don't know nothing until now about the COVID-19 disease.
Okay, another question from João Batista. Thanks for presentation. Is it fair to say that the glycocalyx degradation can explain the higher gravity of SARS-CoV-2 when compared to other coronavirus?
Yes, because the critical patients with COVID-19 disease demonstrated the glycocalyx degradation. So explain the gravity of the disease, mainly in lungs and in the kidneys. I used the endothelial cells. I did not use specific endothelial cells from the lungs or from the kidney, but there are other groups that are studying the glycocalyx degradation in lungs and kidney. And this expanded the gravity of the disease. And also we observed in Ribeirão Preto Medical School that some patients that present the COVID-19 disease, but was mild to severe, they were not in the hospital. They confirmed the COVID-19 patients and they come back home to stay home and wait the steps of the disease. But some months later, they come back to the hospital from Ribeirão Preto Medical School, and they were with the coagulations and thrombosis problems in the lungs since associated to glycocalyx degradation.
Thank you, Simone. Another question from Professor Fernanda Giacchini. Simone, this was an excellent talk when you showed interleucin 6 and interleucin 1 beta levels not served patients. The level were increased. However, he continues the question. However, is it possible to observe that several COVID-19 patients did not show increased cytosine levels? It's like the COVID group displayed two separate profiles. Have you noticed it?
Yes, thank you, Professor Fernando, to your question. We noticed this, we observed this, and the plasma from our patients was collected when after 5 days that the patients were in the hospital. So in the beginning of the disease, but some patients stay in the hospital for 15, 20, 30 days. And when we use the plasma from these patients after the 5 days, we did not observe any difference in interleukin 6 or 1 beta or 10F alpha. We did not observe any difference. So just in the beginning, I did not told this in my presentation, but the plasma from our patients was collected between the 5 beginning days of the disease.
Thank you, Professor Simone. I have a question. How long for the cells to recover the glycocalyx function? Do you think that the post-COVID syndrome could be related to the persistent damage in glycocalyx?
Yes, I think yes. We want to make this perform the treatment with plasma and take out the plasma and check how long it's possible to recover the glycocalyx. But some groups perform the shear stress in endothelial cells, and they observed that Syndecan-1 and Glypican-1 can recover 30 minutes after the shear stress is stimulated. But these cells was in a good conditions, but in conditions with COVID-19 patients, I think there is no study in this moment. So it's a good question, Professor Aline, and I want to keep doing this research, but I need to come back to Ribeirão Preto to do the experiments.
Yes, and another question about the renin-angiotensin system. Since the SARS-CoV needs the heparin sulfate to binding to AC2, what do you think about the renin-angiotensin system as a therapeutic approach? Do you think that this could be a better approach just with association to heparin or not?
I don't know. I really don't know. I think it's very complicated because there is a different pathways to do the experiments and different pathways to the disease. So I really don't know, because the SARS-CoV degrades the SARS-CoV infection degrades the glycocalyx, but also the virus needed the heparan sulfate to attach to the cell. So I really don't know to answer your question. Sorry.
Okay, it's indeed there is a lot of questions to be answered in the SARS-CoV, especially with these new variants too. Probably this shedding on glycocalyx could be more intense in this new variant. Do you think this could be?
Yes, I think so. Because I think the virus is changed his mechanism to attach and to infect the cells, improve the mechanism.
Yes. Any more questions?
I have a question if we have time. So Simone,
Yes,
thank you for your talk. And this post-COVID syndrome is when the symptom persists over 4 weeks from their onset. And then you showed that you took samples from patients from the beginning. I don't remember if you said 5 days or something. And do you have any data on glycocalyx degradation, the participation of the system in post-COVID syndrome patients? I mean, in the long-lasting symptoms, the sequela from the SARS-CoV-2 infection?
No, but we have the samples in the -8 in our freezer in Ribeirão Preto, because we collected the blood in the first day of the patients were in the hospital, in the second, the 5, I think the 7, but we made a scale between 30 days. And some patients died, but in other patients, we made the collection of blood during the 30 days. And we want to keep doing the experiments, but I was in the United States for some months doing a postdoc this year. And now I'm back to Brazil. I have been in Brazil for 2 months, and I want to continue the experiment and answer your question, because I think it's important.
Yeah, I suppose you would follow up these patients. This is why I've asked it. Okay, thank you.
Thank you for your question, Professor Carlos.
Thank you again, Professor Simone, for being here and share your such interesting data. And now we going to the next speaker. And the next lecture will be given by the researcher, Raiany Alves de Freitas. Raiany has a degree in pharmacy by the Federal University of Mato Grosso, Universidade Federal de Mato Grosso, where she's also earned her master's degree in basic and applied immunology and parasitology. And currently, she's a PhD student in biological science at the Federal University of Goiás, Universidade Federal de Goiás, in the Laboratory of Vascular Biology and Histopathology. The title of her lecture is Interleucin 10 Mediates Anti-Contractile Effects of Perivascular Adipose Tissue and Vascular Remodeling Prevention. Raiany, thank you very much for your presence and for your ability to share your research and knowledge with us. Now I'll give you the floor.
Okay, Professor, thank you very much. Good morning, everyone. My name is Raiany. I am a PhD student of the graduate program in biological science, cursing the second year. I will present now my project entitled as Interleucin 10 Mediates Anti-Contractile Effect of Perivascular Adipose Tissue and Vascular Remodeling Prevention. And my advisor is Fernanda Giacchini. So arteries are blood vessels responsible for carrying oxygen and nutrients to all cells and tissues in the body. These vessels are composed of endothelial cells, vascular smooth muscle cells, and extracellular matrix components, including the collagen and the elastic fibers. These components composing three important tunicas in these vessels: the intima, media, and external. Any alteration in the structure or the composition can cause the vascular remodeling, and the balance of these components are responsible for the controlling of the vascular tone and the ideal rhythm of the blood flow. Moreover, surrounding the arteries is a layer of perivascular adipose tissue, the pivot. This tissue is specialized in storing lipids in adipose cells, also called adipocytes. This tissue have important functions, like a protective barrier, energy and thermal source, and endocrine action. There are four types of adipose cells. The white tissue is more associated with the energy storage and the endocrine action. The pink cells is more involved with the milk production during pregnancy and the energy storage. The beige tissue is one association of the white tissue and the brown tissue. And finally, the brown tissue is more associated with the thermal storage because the high number of mitochondria in the cells. In general, the white tissue is more associated with the inflammation because the high release of substances in the endocrine action by the cells, while the brown tissue is more associated with the metabolic activity. It's important to mention that pivot, the white tissue or the brown tissue, can change their phenotype according to the anatomical region, this tissue in the body, or local and systemic stimulation, like a treatment, temperature, and other stimulations. Pivot is also composed of other cells, including immune cells and extracellular matrix components. All these cells, mainly immune cells and adipose cells, can produce and release substances, also called adipokines. These adipokines, such as adiponectin, leptin, growth factors, and interleukins, have different actions and functions in the vasculature and other tissues. In fact, the pivot is a broad source that releases substances directly into the vasculature. In one physiological state, these substances can reduce the contractile response, one anti-contractile effect by pivot, attenuated vascular inflammation, reduce atherogenic factors, and reduce tissue damage. So, in one physiological state, the presence of pivot is beneficial and protective to the vasculature. And one important mediator released by the pivot is the interleukin 10. Interleukin 10 has several essential functions to maintain tissue integrity and homeostasis during physiological process. The cytokine promotes important and protective effects in the vasculature and other tissues. In fact, the cytokine has been used as a treatment for immune disease. However, the role of IL-10 in the vascular structure and composition, and the role of IL-10 as a mediator of the pivot effects on the vasculature remains unclear. For it, the goals of this study was to evaluate the role of IL-10 in vascular remodeling and mediator of the anti-contractile effect of pivot. For it were used male mice with C57 lineage and animals knockout for IL-10 with age 12 weeks. First, we removed the thoracic aorta of these animals and separate in different groups. First, the thoracic aorta without the pivot. Second, with the presence of pivot. And finally, the pivot isolated. Until now, we performed the histology, to analyze the vascular morphometry, elastic fibers, and collagen deposition. Vascular reactivity to evaluate the contractile effect mediated by phenylephrine in the 10 micromolar concentration. And Western blot to evaluate the uncoupling protein 1, the UCP1 expression. That's one important marker of the brown adipose tissue. So in the morphological analysis, the endogenous absence of IL-10 promotes a reduction in the pivot area, as observed in this figure, the reduction in the pivot area compared to control animals. We also observe an increase in the vascular area and in the vascular thickness, suggesting a vascular hypertrophy in the absence of IL-10. In the analysis of extracellular matrix components, we observed an increase in the percentage of collagen deposition, as observed in this figure, using the Picrocereus dye. We also observed a reduction in the percentage of elastic fibers, using the resourcing dye. So in these conditions, in the increase of collagen deposition and the reduction of the elastic fibers can impair the resistance and the elasticity of these vessels. And consequently, these conditions can alter the vascular tone. In the protein analysis, the IL-10 knockout animals demonstrate a reduction in the UCP1 expression in the pivot tissue compared to control animals. The UCP1 is one protein responsible for the heat production in the mitochondria present in the brown adipose tissue. In fact, in the morphological analysis, we observed more brown adipose tissue phenotype in the pivot of control animals. However, in knockout animals, we observed more white adipose tissue phenotype. In fact, in the physiological state, the pivot of thoracic aorta naturally is more associated with the brown adipose tissue phenotype. However, in the absence of IL-10, we observed one tissue more associated with the white adipose tissue phenotype, more involved with the inflammation, frequently observed in some conditions, like hypertension, diabetes, and cardiovascular disease. Finally, in the vascular reactivity, the presence of pivot reduced the vascular contraction induced by phenylephrine in the 10 micromolar concentration in control animals. So that is an anti-contractile effect of the pivot in control animals. It's already well established. On the other hand, the endogenous absence of IL-10 results in an increase in the vascular contraction compared to control animals. So the animals knockout for IL-10 naturally presents change in the vascular structure and composition, and one increase in the expression proteins involved in vascular contraction. So these alterations can impair the vascular contraction in these animals. In addition, the absence of IL-10 results in an elevated blood pressure and cardiovascular dysfunction. However, the presence of pivot in thoracic aorta from IL-10 knockout animals does not reduce the vascular contraction. So these results demonstrate in the absence of IL-10, the pivot does not promote your anti-contractile effect. So it's important to mention the important function of the IL-10 for this effect. In conclusion, the IL-10 plays an essential role in the vascular composition and function by preventing vascular remodeling and mediating the anti-contractile effect of pivot in the vasculature. In the next steps, I will evaluate other important components of the morphological analysis, like adipocyte size and different types of collagen, the inflammatory cytokine profile released by pivot, and important mediators of the extracellular matrix components like metalloproteinases and TAMPs. So all these results will demonstrate the role of IL-10 in the vascular structure, in the vascular function, and in the vascular homeostasis. Thank you to my lab and colleagues. Thank you the universities that I can perform my project, and the development agencies, HIPS and CNPq. Thank you very much.
Raiany, thank you very much for your presence and for your availability to share your research with us again. And now we are open to questions. I think Carlos, do you want that I read, or you read the questions?
It's okay for me to read. Let's see.
Okay.
We have Dr. Rodrigo question, Dr. Rodrigo Gomes. He's saying it's a very interesting your UCP data in the PVA tissue. And I guess the questions come through, how is the adrenergic response in PVAT?
About the receptors, Professor. I don't remember to see about the adrenergic function and these receptors. I'm sorry. But in the functions of the pivot, is interesting to observe the effect of pivot in one physiological state compared to a pathological state. So maybe these components and receptors is different. So it's important to consider the conditions that you think that you make your question.
Yeah, he's extending the questions for beta 1 and beta 3 receptors. Beta 2 and beta 3 receptors, sorry.
Yes, I see. But how I say, I don't remember to see about the adrenergic functions, but and the receptors involved in the pivot. But it's important to consider the conditions that you make your question, because the pivot is different in some conditions. So in physiological state, you have pathological state in diabetes, hypertension, and the other conditions. So I will see it more.
Okay. So next question come from Dr. Diego Colognati. It's a great talk and results. Do you know if in obesity the phenotype of the pivot is different?
Yes, of course. Yes, the pivot is a modulator tissue. So in one physiological state, the pivot is beneficial and protective. Release just substances protective to the vasculature and other tissues. So the endocrine action by the pivot in one physiological state is so protective. However, in the obesity, diabetes, hypertension, the pivot change your structure, your composition, the release the mediators released by this tissue. So is different, completely different, and in all these inflammatory disease and inflammatory conditions, the pivot change.
Okay. Following, Dr. Tiago Costa is greeting you for your very nice talk. And he asks, did you evaluate the mitochondrial function in the NIM pivot of IL-10 KO mice?
No, just the UCP1 expression, because the UCP1 is one marker of the brown adipose tissue, and my goals of this study is evaluate the difference of the phenotype. So one phenotype more associated with the white tissue or the brown tissue. So I didn't evaluate the mitochondrial function, but it's a good idea for the next steps.
Okay, so the host is forwarding a question from Dr. Simone Potje. Raiany, congrats for your presentation. Do you think that in your model, IL-10 could exhibit change in the expression of some specific component of Glycocalyx?
Yes, maybe. Yes, I didn't evaluate these components, but I think it's important to consider this effect, because all these changes in these components, in the structure and composition, can cause the vascular remodeling. So I think it's important to consider it too.
So subsequently, she invites you to do some collaboration. As for if yes, we could test the samples of your animals in Tosta's labs in Ribeirão Preto Medical School. So she's welcoming you to do something together.
Yes, my animals from there.
Okay. Dr. Aline, do you have further questions?
No, just was an excellent lecture. And in fact, this kind of relationship between interleukin-10 and the anti-contractile effect in pivettes, it's a kind of contradictory in the literature. I've been seeing some studies showing that there is no relationship of interleukin-10 in this anti-contractile effect in health condition. But you showed that yes, could be. Yes. Do you think that there is epigenetic condition that could predispose more to this dysfunction in some pathological condition, like obesity, like diabetes? I would like that you talk more about this, your perception that you work with this more.
Yes, thank you, Professor. In the first talk about your sentence, yes, I remember this paper. I don't know, but I think this paper is the Stephanie Watts. I don't know, but Stephanie Watts recently published one paper about the association with the interleukin-10 and the pivette. And she doesn't see nothing difference in this. So IL-10 don't contribute to the anti-contractile effect of pivette in the health. But in this paper, she used the mesenteric arteries. So in mesenteric arteries, have a phenotype more associated with the white adipose tissue. And in my study, I used the thoracic aorta. So thoracic aorta have a phenotype more associated with the brown adipose tissue. So these conditions can change in different tissues and the regions, anatomical regions in this tissue. So the first is it, and I remember this paper too, and it's so nice. And about the model, yes, I agree. The animals knockout for IL-10 naturally presents these changes, these alterations in the vasculature and other tissues and organs. However, in the next steps, I want to evaluate the endogenous function of the IL-10 and the exogenous function of the IL-10. So I think the exogenous function of the IL-10, like incubation with the cytokine in the pivette, in the aortic tissue, can respond better these questions about this model animal. So I think it's necessary more studies to evaluate the endogenous role, and the exogenous role is really important now.
Thank you, Raiany. Thank you very much for your answers.
Thank you.
Is there any more question?
So Aline, while people takes time to Tiago asked some questions. Sorry.
Yes, I think that, I don't know if we've read his question. I think not.
Yeah, I just took it now.
Yeah.
So the question is, in IL-10KO animals, what is the phenotype of white pivot mesenteric arteries? Are there any changes?
Yes, I told you, Professor, recently about it. Yes, in the different arteries of the body, different anatomical region in the body has different phenotype of pivot. So in one thoracic aorta, we have a phenotype more associated with the brown adipose tissue, so more involved with the metabolic activity. However, in the mesenteric arteries, we have pivot more associated with the white adipose tissue, so more involved with the endocrine action and the release of substances, and more associated with the inflammation naturally. So yes, have differences is very important to note this difference in some conditions, because in one inflammatory disease like hypertension, cardiovascular disease, obesity, these conditions can change too. And so, these conditions, inflammatory conditions, this pivot can change your phenotype to white adipose tissue, more associated with the endocrine action and inflammation. So the pivot change according the anatomical region and according the conditions of this patient. I think that's it.
Okay. Thank you, Raiany.
So Raiany, she's having a lot of prizes from the audience, including the program of biological science. It looks like the way you proceed is reflects the quality of the students this program wants to graduate. So congratulations for you and for all the students that have been presented these days.
Thank you very much, Professor.
So now we will take a break of these presentations for everybody. Take a little coffee and be prepared for the next round of lectures. We will return at 10:15 a.m. Thank you. Thank you, everybody, for watching us.
Thank you. Bye-bye.